Thanks.
Okay, thank you. So I'm a PhD researcher in David Richardson's group, and I'm going to be talking about some of my work in the Berthelot pipit. So as it's clear to us, pathogens are a strong selective pressure because they can have considerable impacts on host fitness, and therefore there's often strong host pathogen coevolution. And understanding the forces that drive variation, or the mechanisms that maintain variation and which genes are involved are incredibly important. One powerful way in which we can do this is to investigate the spatial distribution of immunogenetic variation within and across recently separated populations of a single host species that face different levels of pathogen-mediated selection. So today, we're going to test for evidence of pathogen-mediated selection and adaptation at both an individual level and a population level, using genomic data sets from populations of Berthelot's pipit. And the pipit is a small passerine endemic to 3 archipelagos in the North Atlantic. And the pipit is an excellent natural system with which to investigate the evolution of immune genes for several reasons. The first of these is because they show nested patterns of population structure. So we can look at patterns of immunogenetic variation between archipelagos, or we can look at patterns between populations within archipelagos, or even between individuals within a population. The pipit also has a well-known demographic history. We know that they colonized the Canary Islands from mainland Africa about 2.5 million years ago. And then from there, they made 2 separate colonization events to the Madeiran archipelago and to the Salvagens about 8,000 years ago. And since then, we know there's been little gene flow between populations. Most importantly, we've been collecting data from these pipits since 2005, and we know that pathogen pressures within a population are consistent over time, but pathogen pressures are spatially variable between populations. Previous work in the pipit has also found some evidence of balancing selection at the MHC, and also evidence of positive selection at TLRs. So one key pathogen that shows spatial differences in prevalence is avian pox. Some populations we've never observed any signs of pox, whereas in other populations, such as this one up here, Porto Santo, they consistently have high levels of pox, up to 33% prevalence. And the disease is caused by a double-stranded DNA virus, and it's characterized by these proliferative regions that you usually see on unfeathered skin of the bird. So usually on the feet, on the legs, or around the eye and the bills. These photos show relatively mild cases, but you can get more severe cases. And pox has been shown to have severe fitness impacts in various host species. Again, pox cases are relatively mild in pipits, but birds that recover do show signs of damage. So they can show missing toes, misshapen bills, etc. But beyond this, there's not really any studies that look at how avian pox influences immunogenetic divergent evolution. So we had 2 main aims. The first aim is to test for associations between key candidate immune genes and pox infection within populations. And our second aim was to see if we could identify any genomic variation that was associated with pox, but at the population level. So we've been sporadically sampling from the pipit since 2005, and we've been taking blood samples. And we have 780 individuals from 2 divergent populations, so from Tenerife and Porto Santo, that are genotyped at TLR4. We also have 310 of those from Tenerife that are also genotyped to MHC class 1. And then for our second aim, we're going to be using double digest RADseq data. And we have this data for 20 to 22 individuals per population for all of the 13 main populations of pipits. And our pox prevalence data is based on the presence or absence of these pox lesions that we see. And this is based on all individuals that we've collected since 2005. So this is 1,661 individuals. So for our first question, I'm not going to talk too much about the methods, but we used individual level modelling, so GLMs or GLMMs, to look for associations between infection. And just to sum up, really, we don't see any association between variation at TLR4 or variation at MHC class 1 and pox infection. In fact, the only significant predictor of pox that we did see was whether or not the individual also had malaria. And then to our second question. So we used a Bayesian genotype environment association analysis using a program called Bayem. And essentially what this does is it looks for SNPs where changes in allele frequencies are driven by environmental gradients. So in this case, our environmental variable is the different levels of pox prevalence that we see across the different populations. And what's really important is that this analysis can take into account a matrix of population structure beforehand. So this is our results. So after filtering for minor allele frequencies and linkage, we were left with 2,334 SNPs. And each data point that you're going to see on this plot will be an individual SNP. So on the x-axis here is base factor values, and this is just a measure of support for the alternative hypothesis in which the genotype shows a linear association with pox prevalence. And on the y-axis here is Spearman's rank, so this is just the correlation between allele frequencies and pox prevalence. And this just functions as well, it serves to reduce false positives because high base factor values can also result from just single outlying populations. So we considered any SNPs candidates if after 5 runs of Bayem, their average base factor ranked within the top 1%, and also their average Spearman's rank ranked in the top 10%. So if I go ahead and show you those. So we had 14 candidate SNPs, and we were able to locate 8 of those SNPs within annotated genes in the zebrafinch genome. And we were also able to identify a further 4 genes that were located within 10,000 base pairs upstream or downstream of those SNPs. And many of these genes that we identified are involved in intracellular signaling pathways or enzymatic pathways, and many of them have been associated previously with other viral infections in other animal species. So just to kind of zoom in on some of those genes, so our highest ranking SNP was found 6,000 base pairs downstream of HSPA8, and this is an isoform of heat shock protein 70, so it's involved in cellular stress signaling. And previous work has found evidence for its role in the replication of poxvirus. And also there's evidence for its role in the suppression of apoptosis, which would lengthen the amount of time that the poxvirus has for replication. UBE2H, it's involved in the ubiquitin pathways. It modifies proteins for degradation and has been previously linked to herpes virus. SMAD2, it's a downstream effector of the transforming growth factor beta signaling pathway, also capable of inducing apoptosis. And this has been linked to diverse viral infections, so Epstein-Barr virus, herpes virus, Westerdahl virus. So you're kind of getting the idea here. So the pox virus has quite a large genome. Much of its genome is dedicated to encoding viral proteins that help evade the host immune system. And this strongly suggests that much of the variation that we're identifying is in appropriate candidate genes that may play a role in adaptation to pox virus. So just to summarize, we don't find any individual associations between variation at TLR4 or MHC class 1 and pox infection. However, the population level analysis identified strong associations between key loci within the genome and pox prevalence. And these correspond to 12 candidate genes that we know make sense in terms of how we know, makes sense in terms of how pox virus is known to evade the host immune system. But now this gives us a series of candidate genes with which we can go on to further investigate at an individual or population level. So we could again do individual level association analyses like we did before. But one of the things that we would like to do is, now that we know that allele frequencies change over space relative to pox prevalence, we also want to look at how allele frequencies at those candidate loci change over time. And this may reveal the mechanisms of selection. And we've got plenty of museum specimens for which to do this. That's kind of what we're hoping to do next. And with that, I just want to say thank you to my funders and my supervisory team, David Richardson, Lewis Spurgeon, and Alex Su, to past pipit students who generated much of these genetic data sets, and to anyone who's helped in the field collect these pipit samples. And I'm happy to take any questions.